welcome everybody. And first of all, let me share with you that this initiative, this first ICM Science Speech, is part of a project funded by the State Research Agency, a project focused on the international talent attraction and retention. And so specifically with this activity today, what we pretend is to facilitate the networking between people and at the same time to share the knowledge they own with the rest of the institute. And what I can tell you is that this project is going to end in December 2025, but nevertheless what we want is to convert this activity in one structural of our institute. And so just some more information about the selection criteria that we used in order to decide the speakers. And we explained in the call, it is about gender diversity, about nationality diversity, and group people belong to diversity. We did, let me say, some fine, but it didn't depends. Doesn't depend on us. Anyway, we will struggle in the future in order to do even better. And some more information is about the way of working of the speech, that we are going to introduce just with the name and the name of the speech, all the speakers, and at the end of each speech, you are going to give a score of this presentation based on the content and the speech same. And another point is about these questions and answer session. I think that we don't have time, so anyway, what we invite you, since this activity is thought for increasing, enlarging the networking of people, we invite you to contact directly with the speaker, introduce yourself, maybe go for a coffee and comment the speech. And so let me now give the speech to Maria, that is going to explain about this core system that we have. And last but not the least, have in mind that the first three speeches more scored will receive a gift today.
Woo! Woo, 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 woo. Thank you, Stefano, for introducing this first ICM Science speech. We are very happy to have all this room full of people. We also are very happy because we have amazing speeches today, and you will need to vote. But first, how are you going to vote? Let's do a quick test. First, you have to scan this code. Maybe some of you already know this app. It's very easy. Don't download anything. Just scan what is on the screen, and from our technical team, we'll send the voting to your device, okay? So when you have the voting in your screens, you can vote from 1 to 10. And at the end of the app, at the bottom of the page, you will find the button Submit. And then is when you are sending your vote, okay? Now you should have a test on your screens. It's just a test. You are not voting for anything. You can be voting about how amazing is it that we are organizing this very nice event. And don't miss that page, because in the same page, you will have to vote every time after each speech. Anyway, in case you miss it, this QR code will be on the screen at the end of every presentation, so you can vote. Okay? Are you ready? Yeah. Then, okay, then.
And so let's introduce the first speaker. First speaker is Teresa Porta, and she's going to tell us about the importance of the particle microbiome in the carbon cycle. Please, Teresa, come on the stage.
Your time will be on that screen, and whenever you're
Sí, vale, pero bueno, está todo como contado ya. O sea, o sea, mejor si no le dais porque son... Ah, ya. I would like you to come with me to observe and listen to the ocean, a vast blue expanse teeming with life. Beneath the surface, a hidden world exists, full of tiny organisms that play a crucial role to our planet's health. At the base of the marine food web are phytoplankton, microscopic organisms that through photosynthesis use sunlight to capture CO2 to grow and create biomass. When these tiny phytoplankton cells die, they fall to the ocean floor, taking the carbon with them. As phytoplankton, zooplankton fecal pellets, and other debris sink, they form fluffy clumps of organic matter called marine snow. These snowflakes are home to a diverse community of microbes. Those tiny dots you can see in these particles. Tiny organisms that break down this organic matter. Think of microbes as tiny cleaners of the ocean. They help recycle nutrients as they break down particles. Depending how fast they do so, they can either speed up or slow down the process of carbon sinking to the ocean floor. What makes microbes degrade particles quickly? What factors slow down microbial decomposition? These questions are still to be answered. Let's take a closer look to different types of particles and stains to label specific microbes that leave attached to particles. Fecal pellets are relevant particles also colonized by microbes. Particles are nutrient-rich hotspots where bacteria and other microorganisms feed on while decomposing the particle. These microbes can change the properties of marine snow, affecting how quickly it sinks. Some microbes produce sticky substances that help the snowflakes clump together and sink faster, while others can slow down the process. By understanding how these microbes interact with marine snow, we can better understand the ocean's role in regulating our climate. I'm starting my PhD research, and my aim is to explore this fascinating world to uncover the secrets of these tiny but mighty organisms, and by sampling the ocean water, performing experiments in the lab and at sea, and by using microscopy and molecular tools. Although these particles may not be as visually appealing as marine animals, they might play a more critical role in the carbon cycle. In fact, this organic rain sustains the diversity of the dark, deep ocean.
Thank you very much,
Estaba
Teresa.
muy
This was
bajito el
such a good start for the day. And now, it's time to vote. Remember, you need to scan this code on the screen, and you have to vote from 1 to 10 this presentation, and click on the button Submit at the bottom of the page. At the end of the day, we will choose the 3 persons that were the most voted. And as you know, they will have a gift. So the next speaker should be getting ready. Now,
So, have you voted? Scored already? Yeah? So, let me introduce the second speaker. Her name is Berta Pinto. She's going to tell us about what photosymbiosis can tell us about sponge regeneration, metabolism, and metabolome. Please, an applause for
Hi.
Berta. Welcome.
So in marine ecosystems, microorganisms not only live in a planktonic state, as we already seen, but they also live in association with plants, algae, and also animals. One of the organisms that live associated to these microorganisms are sponges that are rich in these kind of associations. But we still don't know how these microorganisms participate in the host functions, for example, in the nutrition. The sponge nutrition is really important in the ecosystem because sponges are filter feeders. So they capture the microorganisms and the nutrients of the water, and through their nutrition, they made available for the benthic organism. Also, the secondary metabolism of the sponge produce a huge array of chemical compounds that are, for example, used for their defense. So understanding the role of these symbionts in the host nutrition will also help us to figure which is the function in the bigger picture. So last year, what we did is we target the photosymbionts. These are cyanobacteria that made association with many of the marine organisms. And in the sponge aerophoba that lives in shallow and well-illuminated habitats, the cyanobacteria are abundant. So to try to hack this system of the photosymbionts and the sponge, what we did is we turned off the light of our sponges. With the light, what we did is we deactivate the photosynthesis of these photosymbionts. We expect that without the supply of nutrients and energy of these photosymbionts, the sponge will need to uptake more nutrients to supply their metabolic demands. We also added another treatment that was mimicking predation, because wounding and regeneration is a metabolic demanding process. So we wanted to increase the metabolic demand of both light and dark sponges. Finally, with this experiment setup, we are going to be able to see the changes in the sponge metabolism and metabolome. So first of all, what we analyzed was the regeneration rate of sponges under dark and light conditions. Comparing the start and the end of the experiment, we didn't show any difference. But if you take a closer look to the different time points, what we see is that sponges under light conditions regenerate faster than the sponges under dark conditions. When we analyzed the metabolism of the sponge, what we did is we collected the water, as the sponges are filter feeders, we collected the water that the sponge was ingesting and the water that the sponge was releasing. With this, we know the concentration of compounds that the sponge uptake for their metabolism. So in the oxygen and carbon, we didn't show any difference, but with nitrogen, what we see is that the control sponges the control sponges here are uptaking inorganic nitrogen in form of ammonia and nitrates, and they are excreting the organic nitrogen. However, when we turn off the lights, what we see is that sponges start to ingest the organic nitrogen, and they become a source of inorganic nitrogen for the surrounding water. As we see a similar pattern in the wounded sponges, what we can expect is that this is due to the metabolic increase of the demands. And finally, you have here a cross-section of a sponge. This is what we call the mesohyl of the sponge. And in the middle, there is the osculum. And this darker zone here is where the photosymbionts are placed, because it's in contact with the light. As you see, there is a gradient of the photosymbiont distribution from the external part to the inner part of the sponge. So we needed a technique to use to see the metabolites that are distributed in this sponge. So we used what is called the MALDI EMS technique, and it's a spatial metabolomics. So what you see here are 3 different compounds that correspond to this small tissue. And here is the external part of the sponge, and here the osculum. So there are compounds that are distributed homogeneously in the tissue, some that are granularly distributed, and some that the intensity and the distribution corresponds to the photosymbionts. This is the kind of compounds that we are interested in. So finally, as the conclusions, what we see is that photosymbionts may be helping the sponges to cope with the first response of the wounding, that both photosymbionts and wounding change the nutrient cycling of the sponge and then the chemical composition of the water, and that may be some compounds that are mediated or produced by these photosymbionts. Thank you.
Thank you very much, Berta. No, you can go. Yeah, and now you know it's time to vote. Again, scan this QR or go back to your mobile phones and give your score from 1 to 10 and click submit. Now the next speaker can be getting ready.
And so, just completed your vote. It's now time for our Brita Bataroy, our third speaker. And he's going to tell us about numerical modeling of gravity-driven Well, the problem is my head, so well, maybe I can put the hat on just for a while. Yeah. So again, the numerical modeling of gravity-driven fold and thrust belts of passive continental margin. So it's your round, thank you.
Okay, so thank you. Good afternoon, everyone. After so much of biology, I'm a sole geologist here trying to prove myself something. So I'm be talking about the numerical modeling of gravity-driven fold and thrust belts in passive continental margins. So let me just start with what are passive continental margins exactly. Passive continental margins, as you can see in this diagram, is the transition zone from the continental crust to the oceanic crust, and there is no movement as such in between. That's why we call it passive. Above them, we have some structures known as the clinoforms. These are actually the deltas which are formed due to the sediments deposited from the rivers towards the seaward side. And in between them, we have something called mobile shales. What are these things? So we know that shales are sedimentary structures or sedimentary rocks which are rich in clay minerals. So how do they become mobile? For that, we need to go to this graph below. Here we can see this is the graph of the pore fluid pressure in the sediments present there with depth. So we can see that the pore fluid pressure initially starts to become subparallel to something called hydrostatic pressure, which is the pressure exerted by the water column from the surface to the depth of interest. Then, After approximately 3 kilometer depth, it starts to become subparallel to lithostatic pressure, which is the pressure exerted by the solid matrix or the solid part of the sediments. And when the shales in this region become subparallel to the lithostatic pressure, we get something called mobile shales, which means the shales start to flow or deform in a manner that they start to mimic flowing. And sometimes these shales move up to the surface to something known as mud volcanoes, which many of us might be aware of. So we are trying to mimic, or we are trying to simulate this entire process which I told in something called finite difference model in MATLAB. So if I just go through the basics, what we do is we define a marker for each material present in the setting, and we try to deform it in a particular direction to mimic the progression of the deltas from the landward side to the seaward side. And we try to track the changes of the markers as they go from one grid to the another. These markers contain material properties such as temperature, viscosity, and the grids remain stagnant. So we can track the changes of the temperature, viscosity, elasticity, etc. as it tries to move from one position to another. So finally, this is my model. And as you can see, this is what we tried to simulate. Initially, we can see that the sediments are trying to move from the landward side to the seaward side, and we have some structures involved here. We have something called normal faulting, which is which is due to the accumulation of the sediments of the deltas from above, and we have the thrust faults or the imbricate fans in the seaward side, which is due to the mobile shale pushing the sediments upward as it goes towards the seaward side. And we can also see these diapirs forming with time, which actually, when it comes up to the surface, forms these so-called mud volcanoes. And this is the strain rate graph. It just shows the movement which is happening in this entire setting. The lower strain rate is depicted by this green color, and you can see it is much lower, which means the strain releases once the movement happens, and it is much lower in the fault and in mobile shales. So this is the final model with our last time step, which you can see is very much similar to what we get in nature in an idealized passive continental margin setting. Last but not the least, we have some positive and negative socio-economic impact related. We have mud volcanoes which are extremely violent, such as Lucy in Indonesia, which has been erupting since 2006, and it has been destroying buildings and people migrating from the place. We have mud volcanoes are a huge source of methane. So they are forming methane deposits which try to sustain many biodiversity or try to sustain the ecosystem in a submarine settings. Also, mud volcanoes are highly associated with oil and gas reservoirs, so they are extremely useful in geo-exploration. Thank you.
Thank you very much. It's refreshing to have some geoscience between all this biology, yeah? So, time to vote. You already know the instructions. Hope I don't have to repeat myself
and
anymore.
again, time to vote.
So, time for the next one.
So now, Sandra Ramirez Calero, our fourth speaker, is going to speak about transcriptomics mechanism for the conservation of Mediterranean octocorals impacted by the climate change. So, welcome, Sandra.
Well, thank you very much for the introduction. I'm going to try to speak with this microphone. So I would like to start by stating the obvious. We live in a world that is constantly experiencing global changes. One good example of these are the marine heat waves that we see annually here in the Mediterranean. We know that these marine heat waves have increased in intensity and frequency in the past few decades. And in addition, they have been associated with even worse effects, such as mass mortality events that affect several taxa, specifically benthic macroinvertebrates, such as habitat-forming octocorals. However, the levels of affectations of mass mortality events are never homogeneous, meaning that their mortality patterns are often contrasting between species, populations, and individuals, generating differential responses. Many hypotheses have been proposed to explain this, but many of them, or most of them, tackle this problem by individual approaches or using specific scales. So the aim of my thesis was to combine different scales, such as temporal, spatial, and taxonomic, in order to identify the underlying factors from a phenotypic, genetic, and ecological perspective. So since I have only 5 minutes, I'm going to give you an example of how we're tackling this problem using the precious coral, Corallium rubrum. So how did we do this? We collected 3 different individuals of Corallium rubrum in 2 different populations in the Catalonian coast of Spain. One considered as shallow and one considered as deep. Each of these individuals was replicated in a common garden experiment that had a control at 18 degrees and a treatment at 25 degrees. Each of these replicates was sampled for RNA sequencing at 3 different time points in order to assess their initial response to stress, mimicking a marine heat wave. So this was day 0, 5, and 10, and this was conducted for 15 days. By conducting several differential gene expression analyses, we observed big differences between populations. If you look to the left, you will see a Venn diagram that is showing the overall transcription of each population under heat stress, and you will see that the deep population has a way larger transcription in comparison to the shallow. So now if you look to the right, you are seeing a dot plot graph that is showing the most significant molecular functions that were being transcribed by each population. And here we not only observed that the deep population had way more functions being transcribed, but it had unique functions such as thermal response, myosin, and collagen, and also some of the shared functions with shallow were having a higher expression. In addition to this, we also found a high variability in this initial response to thermal stress, since the deep population transcribed differential expressed genes in each of the days sample, whereas the shallow only did it at day 5 and 10. Now, if you look to the right, these patterns were very different because the deep population had the highest gene expression by day 5th, and it had a slight reduction by day 10th, and this was completely different for the shallow population since the molecular functions being transcribed were more or less similar in between both days. So as a conclusion, the transcriptomic signatures suggesting contrasting mechanisms among the populations to cope with this stress, with heat stress, in particularly for the deep population that had a unique and higher expression of molecular functions. In addition, the patterns of expression of each population might confer them different predisposition to response to marine heat waves, and this might be particularly lower for the deep population since it's a population that historically has not been exposed to high thermal regimes as a shallow population might have. And therefore, this might be suggesting potential local adaptation depending on the habitat that we are looking at. And finally, findings such as this should be definitely considered when discussing the further differential responses that we often see for Corallium rubrum in nature, and therefore should be definitely considered for promoting conservation strategies when it comes to this species. So, as always, I would like to thank my research group and my sponsors for allowing me to conduct this research. If you find this research useful, please let me know at the end of the science speech in the ICM. And thank you very much for listening.
Thank you very much for your presentation. She still had 20 seconds to go. So now you know, time to vote. You're from 1 to 10 and click submit. And we need some time To rest? How are we doing?
We are at the right pace.
It's very fun. Okay, so continue.
So next speaker, our fifth speaker, Sergi Martorell, and he is going to speak about dormancy in marine bacteria. And so give a warm welcome, Sergi.
Thank you for the introduction. Well, today I'm going to talk a little bit about my thesis project. This talk is called Dormancy in Marine Bacteria, but I'm going to explain mainly two different concepts that I think are needed to understand the project. The first one is dormancy, that's already in the title, and the second one is toxin-antitoxin systems, and then I will talk about how they can relate. So first, what's dormancy? Well, when bacteria are growing in good conditions because they have plenty of nutrients, they have a nice temperature, they are very happy and they are constantly growing, reproducing, they maybe are moving around, but that's not always the case, and in fact it's not usually the case. So a lot of times maybe there are not enough nutrients or they can get infected by phages and they can die. So they obviously don't want to die and have some mechanisms to prevent this from happening. And one of these mechanisms is the dormancy state. That's a state where they change their metabolism, they reduce it to the minimum levels, and they also change their morphology, they reduce their size, and they become much more resistant to nutrient starvation and also to phage infections and other stresses. This state is reversible. They can go back to a growing state if the conditions are good again. They are not immortal. They can also die in this state, but they are less vulnerable. So why is important to understand this state? It's because it has been estimated that up to 40% of marine bacteria can be found in a dormant state. And it's also important because they can act as a seed bank, giving more resilience to the microbial communities and also making them more stable in a long period of time. So that's it more or less for dormancy. And now I'm going to talk about toxin-antitoxin systems. So these systems are genetic systems, conformed of two different genes, very close together in the bacterial genomes. They are found around almost all kinds of bacteria. And one of them is a toxin that, well, encodes the toxin that's toxic for the bacteria that produces it. And the other one is a complementary antitoxin that inhibits the toxic activity of the toxin. So usually it inhibits it by attaching to it. And how is toxic the toxin? So its toxicity, it happens because it targets some essential parts of the bacterial metabolism, such as DNA replication, cell membrane synthesis, or RNA translation, for example. So when this happens, the metabolism of the cell stops, and when the toxin attaches to it, as I said, it inhibits its toxic activity, and the toxin can no longer stop the metabolism. And how is this related to the dormant state? So it has been hypothesized that because of this ability of the toxins to stop the metabolism of the cell, it could be used by bacteria to regulate the switch between this active growing state and the dormant state. So one of the main hypotheses of our project is that a higher number or a higher diversity of these systems in the bacterial genomes could improve the regulation of this switching, and ultimately it would increase the survival of the bacteria in changing conditions or difficult conditions. So to see this, we're going to do some growth experiments, proteomics and transcriptomics, with different marine bacteria such as Alteromonas, Roseobacter, and Doctonia. And well, that's it. Thank you.
Thank you very much. He still had a minute to go. He was able to sum it up in 4 minutes.
That's right.
So, let's give you some seconds to vote this presentation. Now we are in the Ecuador. We are in the middle of this first ICM Science
It is.
So we still have some speakers to go.
Yeah.
Shall we
Share the next one?
Okay.
Why not? So now we are calling to the stage Teresa Morganti, our 6th speaker, and she's going to tell us about removing dissolved organic pollutants from the water with a question: can marine sponge biodegrade or bioaccumulate them? Thank you.
Thank you. Okay, so hi everybody. Today I'm gonna present part of my line of research, which is part of the Batterie de Pinot. And so, can marine sponges biodegrade or bioaccumulate dissolved organic pollutants? So first of all, why sponges? Because they have simple body plan, which is composed by an aquiferous system. They are present everywhere, worldwide, and they filter large volume of water. So just to give you an idea, 1 meter square of sponge community can filter in one day 170 meters of the water column above them. Here you have a video of a Mediterranean sponge, very small, and you can see how fast they pump the water. On top of that, sponges live in association with microorganisms, and they have developed 2 life strategies. One, hosting high, abundant, and dense microbial community in their tissue, called high microbial abundance, and the second one, hosting less dense and less diverse microbials inside their tissue, called low microbial abundant here. So my overarching question is, what is the role of sponges in recycling dissolved organic matter from anthropogenic origins? And so dissolved organic pollutants. And I was inspired by several studies who address the important role of sponges in nutrient and organic matter cycling. Everything started in the 70s, where Riesving addressed the importance of sponges in the benthic pelagic coupling and suggested also dissolved organic matter as a food source for sponges. Only in 2003, Yael demonstrated that dissolved organic carbon can be ingested by sponges after several bunch of enthusiastic researchers demonstrated DOM ingestion in different species and also geographical area. In 2013, there was a study that equated the role of organic matter cycling by sponges to that of the microbial and called the sponge loop. Finally, also, not only the symbionts inside the cells, inside the sponge, but also the sponge cells can ingest DOM. And the last studies, 2 studies, show anthropogenic dissolved organic matter from sponges. So this was what inspired me to address this question. And I hypothesized that sponges are able to take up anthropogenic dissolved organic matter. They may also bioaccumulate them or degrade. And also they may process ADOM differently due to the different life strategies. The goals are to explore ADOM cycling processes by filter feeders and possibly use sponges as bioreactor models. In order to do that, we had an experiment set up here in the ZAI, and we exposed the sponges to a fixed concentration of dissolved organic pollutants. In detail are polycyclic aromatic hydrocarbons and orthophosphoric ester. And we collect with a vacuum system the water inhaled and exhaled by the sponges in order to estimate the possible ingestion or excretion of different compounds. And this is part of PhD thesis from Nuria and master student Aitsiber. So both they will be working in the ZAI. So if you want to pass by and see the setup, you're welcome. And so these are all the planet analyses, just to give you an overview. And so, of course, we will measure anthropogenic dissolved organic matter, but also bioaccumulations, and also the symbionts inside the sponge tissue in order to check whether there are bacteria that are known to degrade dissolved organic contaminants in the sponge tissue. And then we have different collaborators from IDAEA and also ICM. And finally, I would like to thank all the collaborators, but especially also the funding agency, the AGAUR, otherwise I wouldn't be here. Thank you.
Thank you.
Thank you very much. Now you know, time to vote. And now, as every event needs, we will have some technical issues.
Of course.
Because every...
The head inside. That is the issue. Anyway.
So, please, we...
Slide.
Yeah?
So let me introduce Hector Perea, our seventh speaker. He's going to speak about earthquakes from the ocean depths, exploring offshore faults and their impacts. So, now, just a moment.
Yeah, you can see as I'm an H outlier here, so I'm also have some deficiencies in interesting videos on the screens or on the presentations, but don't worry, I will try to do it.
Yeah.
Nicely.
It's time.
So, yeah.
Welcome, Hector.
I hope it was fast enough. So what I'm going to talk about today, it's about active faults or offshore faults that are faults that can be capable of producing large earthquakes and tsunamis in the ocean. So maybe you are wondering why this guy is studying active faults offshore if nobody lives there. Yeah, maybe it's true, nobody lives there, or almost nobody lives there, but these faults can produce a shaking, a ground motion so high that can produce destruction, that can affect buildings, infrastructures, and also it can produce tsunamis. And both effects can produce high numbers of casualties, deaths, and also at the same time can produce large economical costs. So understanding the offshore faults and characterizing these tsunamis and these earthquakes can allow us to produce or to work on new strategies to mitigate their consequences. So, no, it's the next one. Yeah, here. So maybe you are wondering, everybody here knows, but what is an earthquake? So an earthquake, an earthquake, you have a fault. Along this fault, there are some energy that is accumulating. We call it stress. So, but there is a friction between these blocks that when this stress just go above this stress, this friction force, it produces a displacement where the fault deploys all the energy producing the earthquake. And what about the tsunamis? More or less, the tsunamis is the movement of this fault on the offshore, on the water, and when the seafloor is displaced because of the movement of this fault, it produces a wave, a tsunami wave. These waves are on the offshore. They have long wave distances, but they have a very short amplitude. But when they arrive to the shore, due to the friction with the seafloor, they increase the amplitude and reduces the wavelength. So they produce high waves that can flood very large areas. And how we explore this? Of course, these faults are quite deep on the ocean, so we cannot dive down there. So what we do, it's like we go with large vessels to the offshore to acquire and to deploy the different equipment as ropes and IOVs, and acquire diverse data. And with this data, some of the data that we use, it's bathymetry. Bathymetry produces the maps of the seafloor, and in these maps we can recognize different features, geomorphological features, that are giving us some information about the faults, as for example, alignments or tectonic basins or parallel small scarps or here like channels displaced by a fault. But also, we work with seismic data, with the seismic data that gives us this information about what are the geological structures that are below the seafloor. No? So in that case, in these profiles, they have different resolutions. As you can see here, there are some parallel lines that we call reflectors, and they are representative of the sedimentary layers that we have on the seafloor. And at the same time, in here, you can also see that there are some vertical lines that are displacing these reflectors. So these are the faults that are moving. And here, what we can see is the cumulative movement of these faults, the repetitive earthquakes. And with all this information, just go. So with all this information, what we can do is to characterize these faults, to get the maximum earthquake that they can produce, to have the time between these large earthquakes, to know more physical properties of these faults, and to produce 3D geological or 3D fault maps, as you can see here. It's too fast. And with all this information, what we get, it's seismic hazard maps or tsunami hazard maps, as these ones that showed that have been done for Europe, but also we can model different historical or recent earthquakes in order to know how this wave is propagating and how the earthquakes, to see what will happen on the future. So just to finish, it's like faults are there, we need to know them, they are going to produce earthquakes, but what we have to do is to study the faults, to characterize them in order to take strategies to mitigate their consequence. And thank you. And now the fast version.
Thank you. Wait, you went too fast. Go, go, go. And maybe if you look at your mobile phones, the voting is already there. But if you need to scan the code again, we need to wait until the slide. Okay, here it is. I've seen that we have some people online. Let me say that the people that is connected online and is watching this on streaming can also vote. So I hope they're voting as well. And we will do a warm welcome to our next speaker.
So we move to our next speaker, Anna Manrique. And so she's going to share with us this topic, the major climate actions begin in the seafloor. So Anna, please, come up to the floor.
So hello everyone, I'm Ana Manrique. Is it working, the micro? Yeah.
Just close to the mouth.
Okay. I'm part of the seafloor and subseafloor geological processes laboratory and of the ecology of marine microbes group. And today I'm gonna present what is the trailer of my thesis. Well, I would like to start by talking about the main focus of my thesis, what is methane. Methane is the second most abundant greenhouse gas in the atmosphere after CO2. And it has the greater greenhouse potential over a period of time. It comes from both natural and anthropogenic sources. And maybe when you think on that sources, you think on ruminants, fossil fuels, wetlands, fresh water. But what maybe you're not aware is that the oceans represent a huge reservoir of methane on Earth. And most of this methane is stored in form of ice, stored in form of these ice-like structures that you see in the pictures, known as gas hydrates. Gas hydrates are rimming the continents, the continental margins, and are only stable under a specific condition of pressure and temperature. What it means that if this condition shift, all this methane that is stored in the form of gas hydrates can be released to the sediments and reach the water column, and even the atmosphere. But before we get so dramatic, let's see what says the last report of Intergovernmental Panel on Climate Change. What I want to highlight in this picture, it's really complex, I know, is that although gas hydrates represent a huge reservoir of methane on Earth, only a tiny part of this methane reaches the atmosphere compared to other sources of methane. Other sources of methane, like fossil fuels, livestock, freshwater, or wetlands. And the reason that a really tiny part of this methane reaches up the atmosphere is that microorganisms are acting as a biological benthic sink, consuming 90% of this methane. But these microorganisms don't do it, but the way that these microorganisms consume methane is not by a single organism doing this, but through a scientific partnership in which anaerobic oxidation of methane is carried out by archaea, and the sulfate reduction is carried out by bacteria. And also, other electron acceptors available in the subsea floor can be involved and coupled to the methane oxidation. In the South Shetland Islands in Antarctica Peninsula, gas hydrates have been detected through seismic surveys. And the ice sheet mass loss that is happening currently can trigger the release of amounts of methane that are stored in the form of gas hydrates. What I'm going to do in my thesis is to take samples of the sediments to look for the signatures of the microbial oxidation of methane. First, I'm going to study the diversity and metabolic potential of these microbial communities. And how is the structure of this scientific relationship? I also use lipidic biomarkers to assess if methane is being consumed or produced. And the main goal of my thesis is to link changes in geophysical trace of the sediment with changes in microbiology, with the microbial communities being in the sediment. And this link between geophysics and microbiology is what makes my thesis a truly multidisciplinary research project. Thank you.
Thank you very much. I hope the public is already voting. You know, you can...
few moments
a few more seconds for you to do
score.
your homework. And I'm very sad because this is almost coming to an end. But we still have two amazing speakers to go. So please, the next one, you can, present.
Thank you, Marie. So the next one is Cynthia Vieira, our ninth speaker, and she's going to speak about revealing secrets of a microbe that wants to be something else.
Hi. Well, what if I told you that the cells of every plant and animal on this planet, including you and you, originated from merging different species of tiny microbes that long ago, millions of years ago, lived separately? This was the beginning of the eukaryotic cell, which came from a series of endosymbiotic events where species A starts living inside a species B. This event took place about 1 to 2 million years ago, and while we have some clues, the exact details of how it happened are still mysterious. In nature, microbes interact in countless ways, but only a rare few interactions have led to the rise of more complex cells. And one of the most fascinating examples is the story I'm gonna tell you. But let me take you millions of years back in a primitive ocean. Actually, it was not that far from today's ocean. It was blue and full of microbes. At the time, there was a microalgae whose name I'm not going to reveal yet. This microalgae, as others, was happily living thanks to the energy of sun. But suddenly, the ancient ocean was disrupted when unexpectedly an asteroid hit the Earth, triggering a period of darkness and a huge decrease in the availability of nutrients. It was the crash of this bolide what caused the extinction, the massive extinction from giant sea creatures to microscopic beings, leaving the once vibrant ocean devoid of species. In the absence of light, these microalgae, in the absence of light, this microalgae was no longer able to produce their own food. However, this microalgae has a hidden ability to hunt and eat other microbes, a strategy that, of course, used to survive. And then something far extraordinary happened, something that we're gonna call the null hypothesis. And here it goes. This microalgae, driven by hunger, engulfed a super powerful bacterium. This bacterium has the unique ability of converting the nitrogen gas from the atmosphere into ammonium. This is not making wine out of blood, but still a crucial compound for all living beings. So right after this microalgae engulfed this bacterium, it started sensing and smelling the ammonium produced, recognizing a vital source or a vital resource for it. On the other hand, this bacterium found a source of food in this microalgae. So this unexpected exchange allowed them both to survive in an ocean with very little nutrients, allowing them, eventually shaping their future. Over time, both started experiencing certain changes in their bodies. Both partners started to divide and to grow together as a single entity. And as a result, there was another thing. This microalgae, one of these changes was that this, in particular, this bacterium, lost some of their functions that allow it once to survive independently in the ocean. So this microalgae with the bacterium transformed it into a more complex cell, a microalgae capable of produce ammonium, a vital resource for all complex life or for all living beings. This bacterium is known as Eucine A, and this microalgae that we named Rhadosphaera live widely distributed through the global ocean. This endosymbiotic relationship opens a window to the past to explore how two organisms or two species could evolve into a single entity. So during my thesis, I'm gonna try to reveal some of these mysteries. But how? Well, the genome of any given species is like a book with the instructions of any species. So I'm gonna use molecular methods to print and read the book of this symbiosis and try to compare them with the book of other free-living species to try to give or provide information about the changes and adaptations that happened from free-living species to symbionts. Thank you so much.
Thank you very much. I'm sad because it's going to end, but I'm
it's
happy
almost over.
because these people is very creative. So
Yeah,
it's been
they
a really
are.
good morning today. So I hope you're already voting, and if you are not,
The
let's
moment
do it.
when you were saying sorry, I have one more thing to say.
And-
Those were like on the stage, or you prepared those from before?
You cannot vote.
What?
So now in the
you
technical
could, Hector,
room, they are
all the
saying
people with the same problem.
that they. So everything is going on. I'm sorry.
You could.
Okay, it's
for
working. It's
all
going
the
on.
speakers.
From the technical room,
Nobody
they're
more
saying
with
that they
the same
have the
problem.
votes for you. So now it's
And now,
last turn.
our last but not the least speaker, Marina Briones, on the stage, and she's going to tell us about the Plastic Empire Strikes Back.
Well, good morning. I guess that from the title of my presentation, you can all guess what is it about. But let me start where everything began. So when I was a child, I didn't know that I wanted to be a marine scientist. In fact, I wanted to be a doctor and an artist. And today I think that it all falls into place because I'm working to be a doctor, not in medicine, but in marine science, and because all my drawings were marine related. Like, how didn't I see it before? Maybe it's because we lack of role models, but that's another topic. So, what is my PhD about? As you have guessed, it's about plastic, and specifically, I study the compounds that plastic release into the seawater. And to do so, I simulate the degradation conditions that occur at the ocean, and I incubate plastic pieces with seawater and I irradiate them, and then I study the compounds that are released. And these compounds can be found in the form of gas, which are named volatile compounds, or they can be found dissolved in the water, which are known as plastic leachates. And understanding the impact and quantifying these compounds is crucial. So last week, my supervisor, Cristina Romera, sent me to the States to convince the president of the Empire State, of the Plastic Empire, about the importance of this research. I didn't went to China because the king of the empire, of the Plastic Empire, was on holidays. So then I traveled, and as the president is a super busy man, I went straight into the point, and I told him our first result, and I told him, oh my God, Mr. President, plastic leachates reduce phytoplankton, organic matter, and production and growth. And I showed to him this growth curve, which is one of our phytoplankton species we tested, but we have observed the same behavior for the majority of the phytoplankton species. And as you can see here, the number of cells per time was lower when the cultures were cultured with plastic leachates, the dark lines, than in the control treatment. And you know what he told me? I couldn't care less. So then I told him, but Mr. President, maybe you are more interested into the volatiles. And I told him that we know that plastic release volatile compounds, and some of them are greenhouse gases, such as CO2 or methane, but we don't know how or how much of them are produced. And besides, there are many other compounds that have not been identified, and that they might interfere with the sensory signals of many marine fauna. So what? So I said, well, Mr. President, you know, phytoplankton is responsible for more than 50% of the world's oxygen production, and it's the ocean's largest carbon fixer. We have seen that plastic leachate reduce its growth, and also it reduces the capacity to fix carbon. And plastic leachates and volatiles can have many other impacts that we don't know on many marine fauna that we depend on. I still don't care. Well, then I told him, well, a reduction in carbon fixation because of a reduction in, or a negative effect on phytoplankton, coupled to increase in greenhouse gases produced by plastic, can have an unknown impact at a local or even global level, and we don't know what is happening. And besides, studying plastic can help us understand, studying the compounds that plastic release can help us understand how plastic degrades in the ocean. Climate action, it's even if it affects someone, it affects everyone but me. So then I told him, look, Mr. President, you are too affected by climate change. For example, that could mean that you will no longer be able to eat lobster or travel to paradisical places. End of the conversation. He said no more, but I think that he was thinking something. So, as the plastic empire strikes back, we will keep working to provide more evidence to defeat the dark side. I would also like to thank Cristina Romera, Rebecca Mañenco, Berta Sala, Nagore San Pedro, and Alex Campillo for their help.
Thank you very much. Time to vote. And don't go far away, because now all the speakers should come to the scenario. Let me remember, this is a contest, and you, the public, are choosing, but with your votes, who is gonna have a gift. We have 3 gifts. So we will have 3 of these speeches, vote that will have this gift. No, the third that had the most votes, then the second, then the first. But yeah. Of course, we all enjoyed every presentation, and everyone should get a big applause for you.
Yeah. You did all. You did, the guys, all. I mean, amazing. And yeah, so we are sure that we are going to repeat this experience, since it's a way, you know, to know better each other and start doing our networking and so on. So Maria, I think that we can invite all the speakers on the stage,
come
please.
up to the stage. All the speakers here. We need to take a group photo. Come on.
So again, Cynthia, Marina, Sandra, please. There's a little bit more. Sergi. Take care, because otherwise, if I fall down from the steps. Rita Bata, Teresa, Hector. Berta, Teresa, my fault. Anna.
We're missing one.
Yeah.
Okay. We can put her with Photoshop if she.
And so now now it's time for the final results. And so there are some folders that are going to come here from the backstage. So we are waiting for them, and we are waiting also for the gifts.
Yeah, are we ready? I'm asking to the technical room.
They are.
I'm asking to our colleagues, Elena and Maria. Yeah, they are ready.
Yeah. Also, an applause for Elena, Maria, and Charlie for doing this event a reality.
Okay, perfect. Look at the gift.
So there we are medals,
It's an amazing.
and we are tangible gifts.
Well, but the important one is this.
Yeah,
we know.
sure. Anna, hey!
Okay, let's make them suffer a bit more. How are you, the public? Did you have fun? Did you enjoy? Did you learn? Okay, no more suffering. We will go from the third to the first. So we win. Okay. We win. Okay. So you have been voting. And now the third person that got the most of the votes was Cynthia Vieira.
Congratulations.
Now, in the second position, we have Marina Briones.
Congratulations.
And please do some drums. In the first position, we have Teresa Porta. Thank you all. Thank you very much. I will ask for a final photo. Please, the three that have the gift, please come here. And you all together here. You follow Charlie's instructions for the photo. And wait. Yay, dale la vuelta. Okay, nice. So I think this was a very nice experience for everyone. I'm amazed about all this. So many good people, speeches, creativity. It was amazing. And then we may have to repeat this next year.
We'll do our best to make another one, you know. So we really, what we need is, commitment by the people like today. We are sure that we are organizing more and more this kind of activities. And at the end of the day, we are very happy about, the development of the event, the participation, and these amazing speakers. And thanks for.
I hope you are all inspired to present your proposal next year. And let's give a final applause not only to the speaker but also to the public. Thank you very much.
Woo!